nervous system characteristics. Okay. Nervous system controls and adjusts the activity of the body, provides a swift but a brief response. So it gives you an immediate response, but it's usually not long-term. Okay. It provides instant reactions to something, but then it goes away. Okay. Long-term responses are your endocrine system. Gets in the bloodstream, goes to target organs. Those are long-term responses. Okay. Swift, short-term responses are your nervous system. Okay. The nervous system includes the central nervous system and the peripheral nervous system. Just so we have the axial and appendicular muscles and skeletons, we got a central nervous system and peripheral nervous system. The central nervous system is the brain and spinal cord. That's what makes up the central nervous system. So you have the brain and you have the spinal cord. Okay? The peripheral nervous system is everything else. It's everything outside the central nervous system. So central nervous system, the brain and spinal cord, responsible for integrating, processing, and coordinating sensory input. Okay, so it receives information of what's going on to the body and the external environment. It's responsible for integrating, processing, and coordinating muscle output. So it takes that information that's coming into it and it decides what to do. Okay? It thinks about it, learns, and then it acts on that input. Okay? It's the seat of intelligence. Memory, learning, emotion are all tied into it. Okay? The peripheral nervous system, remember that's everything outside the brain and the spinal column. The peripheral nervous system provides sensory information to the central nervous system. Once that information is voted on or decided what to do, then it carries those motor commands away from the central nervous system back to the peripheral nervous system. So the peripheral nervous system provides input, the brain decides what to do with it, and then provides output back to the peripheral nervous system. The peripheral nervous system is divided into two divisions. Okay? There's an afferent division and there's an efferent division. Afferent is information coming into the central nervous system. Okay? So all your stimulus, everything in the external environment, proprioception, everything from your body and external environment coming into it is your afferent division. Your efferent division is what it does when it receives that information. Those are the commands that come out of the central nervous system. So efferent is information going in. I said it backwards. I'm sorry. Afferent is information coming in to the central nervous system. Efferent is information going out of the central nervous system. Commands to make your muscles move, make glands sweat, different productions from the endocrine system, things like that. So your two divisions are afferent and efferent. So here's just in case you didn't get it at first. Central nervous system is brain and spinal cord. Peripheral nervous system is everything else that feeds into it and then commands out of it. Okay? So everything outside the brain and spinal cord is your peripheral nervous system. Okay? Like we said, the peripheral nervous system can be divided into two divisions, afferent and efferent. Okay? Here's your afferent coming in, efferent information coming out, going out to. Okay? Afferent brings sensory information towards the central nervous system. Well, this brings it in towards the central nervous system. Okay, and the afferent is divided into two divisions, divided in somatic and visceral. Somatic is your muscles, your voluntary muscles, your control. Visceral is your organs, your glands, things like that. Okay. Efferent carries demands away from the central nervous system. So afferent is going towards, efferent is going away from. And the efferent is divided into two divisions also. The somatic nerves, which are your muscles, and your autonomic nerves, which is digestion, heart rate, pupil dilations, things like that. Okay. These are the basics. So far, so good. Okay. Peripheral nervous system, like I said, divided into two divisions, afferent and efferent, somatic and visceral. Efferent was somatic and autonomic. Now, the autonomic nervous system, you can divide that into two divisions also. That's your sympathetic and your parasympathetic. This is your fight or flight response. your ability to stand your ground and fight, your ability to run like heck. Okay? So that's your fight or flight response. And the sympathetic, parasympathetic is your relaxation response. Digestion, when your body's relaxing, things like that. Okay, so you got afferent and efferent. Afferent is somatic and visceral. Efferent is somatic and autonomic. Autonomic is divided into sympathetic and parasympathetic. And there is a chart coming up soon that'll show exactly what that is that you can use for reference. So we talked about afferent and efferent, the peripheral nervous system. Afferent, the somatic portion, is your skeletal muscles and joints. That's your somatic portion of the afferent system. That's what's coming in or towards the central nervous system. Visceral portion monitors your smooth muscle, cardiac muscle, and internal organs. Okay? So somatic is skeletal muscles and joints. Visceral is your smooth muscle, your cardiac muscle, and your organs. Okay, the efferent we talked about. Efferent also has somatic nerves and autonomic nerves. Somatic is your skeletal muscle, contractions, because remember, this is information going out, so it has to have an effect now. That's the brain decided what to do. So somatic nerves control your skeletal muscle contractions. It's making something happen. Okay? The autonomic nerves control your internal organs. Okay? This is your parasympathetic and sympathetic responses to different situations, fight or flight, things like that. Okay, these are your two divisions of the autonomic nervous system. Okay, your parasympathetic and your sympathetic. Parasympathetic, we'll go over this in a lot of detail coming up. We'll call it all the different effects. Parasympathetic causes your pupils to constrict, decrease heart rate, and it tenses the urinary bladder. Those are just a few of the things it does. Like I said, we'll go into this in great detail. The sympathetic nerves cause pupil dilation, increase heart rate, and relaxes your urinary bladder. The sympathetic nervous system, this is your flight or fight response. This is what your body does. This is what your body does under pressure. Okay, you get in a stressful situation where you have to fight or run, your pupils dilate. They dilate so you can see danger far away. You have more focus ability, your pupils dilate at a greater distance. So they dilate so you can find danger at a further distance. Increase heart rate, provide all the blood and oxygen to your muscles so you can start moving them to fight or run. And it relaxes your urinary bladder. The old expression, when someone gets real scared, they urinate. That's exactly why. Because the sympathetic nervous system is activated. It relaxes your urinary bladder. Okay. Here's the chart I was talking about. If you ever have questions, just refer back to this chart. It's actually a really good diagram. Okay. So this is the two divisions, your central nervous system, your brain and spinal cord, and your peripheral nervous system. Okay. You got sensory input coming in. This is your afferent division. Afferent has somatic, which is your skeletal muscles, your voluntary control, and it has visceral, which is your internal organs, cardiac muscles, things like that. That's input coming in. Then your information is processed. It goes to the cerebrum. Okay? The brain decides what to do with that information that's coming into it. And then it goes out. So this is your efferent division coming out. Efferent also has somatic nervous system, which is your skeletal muscles. Okay? And has an autonomic nervous system. Autonomic nervous system is divided into parasympathetic and the sympathetic division. The sympathetic is your flight or fight response. The parasympathetic is your relaxation response. Okay? The parasympathetic and the sympathetic control your smooth muscles, your cardiac muscles, your glands, causing you to sweat, your heart rate to go up, pupils to dilate or contract, digestion to occur, urination to occur or not occur. Okay? That's what controls your internal effects. So here, this chart is something that if you ever have questions, it's good to refer back to this chart because it explains everything right there of how the nervous system works in a nutshell. All good so far? I don't see any questions. You can also ask questions on the chat. I also got that window open also too, if you have any questions, because questions are good. That's how you learn. Okay, the neural tissue is what makes up the central nervous system and the peripheral nervous system. Right? It's composed of two types of cells. It's composed of neurons, that you talked about a little bit, and neuroglial, which you haven't talked about yet. Neurons. Neurons are the actual cells that transfer or process information in the nervous system. That's where your electrical impulses or your transfer of that information is propagated through, is the neurons. They consist of a soma, an axon, and dendrites. Remember we talked about those last time. The soma is the cell body of the nerve cell, of a neuron. Okay? The axon is where the information is sent down to another cell, or to a muscle, or to a gland, wherever it's going to. The dendrites are what receive information from the external environment or from other neurons. Okay? So basically, I mean, there's a lot of exceptions, but basically you have a cell body and you have all these little dendrites, little tree structures. And all these little dendrites receive information coming in. Okay? Then there's an axon coming out, which is really thick. And this, once it reaches the threshold, triggers an impulse to travel down, an electrical impulse. And it travels down to another cell or muscle. Okay? So this is your soma, your cell body. These are your dendrites, and that's your axon. Great picture, huh? All right. So those are your neurons. Those are one of the cells that make up your neural tissue. You also have what's called neuroglial cells. Neuroglial cells are what support the neurons. They give it its nourishment. They give it the ability to insulate it, to help protect it, to destroy pathogens that will affect your nervous system. Okay, so the neuroglia are supporting cells. Okay, and they protect the neuron. So they get rid of pathogens and they support by bringing nutrients into it and prevent certain things from passing into it. Like the blood-brain barrier, which we're going to talk about in a little bit too. Helps protect the actual neurons by disallowing certain materials to pass into the brain, into the nervous tissue. Okay. Well, this is a much better picture than what I drew. Okay, so these are your dendrites, all these little things right here. All these little tree-like structures are your dendrites. Information comes into those dendrites and it feeds into the soma, the cell body. Okay. Once enough information gets to a certain threshold level, the axon is triggered and an electrical impulse travels along this axon. Okay, so those are your dendrites. Soma is your cell body. Okay. Cell body contains your nucleus, mitochondria, ribosomes, different organelles that we talked about before, Golgi apparatus, endoplasmic reticulum, all the stuff we learned about. The axon conducts the impulse towards the terminal. These terminals here, okay, is where the axon meets either another cell, meets a dendrite, meets a muscle, meets a gland. There's all kinds of terminals it can have, and the synaptic cleft. But it's a synapse. It's a connection between two objects at the end of the neuron. Okay. Okay. So let's talk about the neuroglial cells. These are ones that protect and support the neurons so they can keep functioning. Okay? The neuroglial cells provide the framework for the neural tissues. They're supported. They maintain the intracellular environment. They deliver nutrients. They take away wastes. So they deliver oxygen to the cells. They take away carbon dioxide. They maintain that environment. They act as phagocytes. Phagocytes will eat or dissolve pathogens, bacteria, viruses, things that get into the body for the neurons that aren't supposed to be there. Now, they have the ability to reproduce. Remember, we talked about this. Nerve cells and muscle cells lack the ability to reproduce. The neuroglial cells that support the neurons, they can reproduce. So you can get reproduction of the neural cells, and that's how we can get some nerve repair. Like we talked about before, when wisdom teeth are taken out and they cut the nerve by mistake or damage the nerve, this is how it can be repaired. It gets repaired by these neuroglial cells, not by the nerve cells themselves. They can't reproduce. But the neuroglial cells create an environment where that nerve can grow back into it and reestablish a connection through the neuroglial cells. Okay? So neuroglial cells can reproduce and can have minimal repair of nervous tissue. Not a lot, but it can help repair some. Okay. There's different neuroglial cells for the central nervous system and for the peripheral nervous system. There's 4 types in the central nervous system. There's two types in the peripheral nervous system. So neuroglial cells of central nervous system consist of astrocytes, oligodendrocytes, microglial cells, and epidermal cells. Neuroglial cells in the peripheral nervous system consist of satellite cells and Schwann cells. Okay, we'll talk about each one of those in detail here. Let's see what each one of these does. Okay, this is a summary. These little charts are actually really good because it goes down and reviews. It's an easy reference for you to look at. So these are your neuroglial cells. Remember, neuroglial cells can reproduce, and their function is support and maintenance of the neurons so they keep functioning correctly. That's what they do. They're found in the peripheral nervous system and central nervous system. Okay? Peripheral nervous system, there are satellite cells. These satellite cells surround nerve cells. They maintain oxygen, relieve carbon dioxide, provide nutrients, maintain neurotransmitter levels in the neurons and the ganglia. Schwann cells. Schwann cells, okay, surround all the axons. They're responsible for myelination of the peripheral axon, and they participate in repair after the injury. So say that nerve is damaged by removal of wisdom teeth. Now you got a numb face. Okay? That can last anywhere from 2 to 3 weeks or up to 6 months. Or it can be permanent. If they actually sever the nerve and cut it, there's a good chance it could be permanent. Okay? But if the nerve is just damaged or compressed, there's a chance these Schwann cells can invade the area, reestablish tissues in that area, make a matrix, and that nerve can grow back into that matrix, not make a new nerve. Nerves cannot regenerate. Cannot regenerate. Yeah, they cannot duplicate. Okay, but it can grow into an area provided by Schwann cells and help reestablish a healthy connection. And that's how nerve damage has minimal repair capabilities through these Schwann cells. Okay? Those are your peripheral nervous system. The central nervous system, okay, has the 4 types of cells. They have oligodendrocytes. Oligodendrocytes maintain the central nervous system, and they provide the structural framework. They're like the connective tissue that holds them together and keeps the nerves where they're supposed to be. They support the neurons in their structure and allow them to do their thing, to transmit information. It has astrocytes. Astrocytes, the big thing with astrocytes is they maintain the blood-brain barrier. The brain is very sensitive. Okay? Certain things cannot get into the brain or else it will destroy that tissue. And this is what keeps them out, these astrocytes. They create a blood-brain barrier for the brain. Okay? They provide support, regulate ions, nutrients, dissolve gas concentrations, absorb and recycle neurotransmitter, and form scar tissue after there's an injury. Okay? Are the functions of the astrocytes. You also have microglial cells. Microglial cells are your phagocytes. Okay? Those are the ones that eat bacteria, pathogens, viruses, things that don't belong. These are things that dissolve it, make sure it doesn't continue to damage any of your nerve cells. And then epidermal cells. Epidermal cells, they line your ventricles of the brain. We'll talk about it in a little bit. The brain has fluid to it with these little ventricles that help support it, provide nourishment to the brain cells. Okay, it lines the ventricles in the central nervous system and spinal cord, producing, circulating, and monitoring of cerebral spinal fluid. So the epidermal cells monitor your cerebral spinal fluid. Cerebral spinal fluid is very important in keeping the nerve cells healthy. Okay? This is what bathes the brain tissues and the rest of the neurons in the central nervous system and keeps the nutrients and everything around that's supposed to be. Keeps everything balanced correctly. Okay? I think we'll break it down and doing each one of these. So central nervous system. One of them is the astrocytes. Astrocytes have a large number of cytoplasmic processes. Okay? They control the chemical content of the interstitial environment. They keep the oxygen level up so oxygen can transfuse into the cell. They keep the CO2 level down by getting rid of it so the waste products can come out of the neurons, okay, by diffusion. Maintains the blood-brain barrier. That's probably the most important part of astrocytes is they maintain the blood-brain barrier. So they isolate the neurons from the general circulation. Okay, your nervous system is not exposed to your general circulation. Now think about what that means. It means all your antibodies and everything that your body recognizes as self never gets exposed to the central nervous system. So if there is a disease or a breakdown where all of a sudden the central nervous system is exposed to your general circulation, you have an autoimmune disease. You have a disease that's going to start breaking down your body because it thinks it's foreign. It was never exposed to it when your immune system was developing because it's a separate system. So if something happens and it gets exposed to it, now your body's going to recognize that as foreign. So your antibodies are going to attack your central nervous system, therefore giving autoimmune disease. It recognizes your own body as foreign. So these astrocytes maintain a really important role in keeping this blood-brain barrier isolated. It keeps your central nervous system isolated from your general circulation, your antibodies and everything else, because your body will destroy it if it sees it. Okay? That's why if you have herpes, okay, cold sores in your mouth, herpes, like some part of that, they live in the central nervous system. That's why if you have herpes, you have it for life. There's no way to get rid of it because it can't go into the central nervous system and destroy it. That's why it comes in the same place every time. It always sends the same place on your lip, same place in your mouth. I get canker sores at the base of my tongue. Hurts like crazy. Comes to the same place every time because it lives in the central nervous system. And when it gets activated, it travels through that same nerve path and comes out in the same place. That's what's going to occur in the same place. And that's why you can treat it, the symptoms, but you can't treat it and get rid of it. You're going to have it for that because it lives in your central nervous system where your body is not going to find it because it's not exposed to it. Okay? It's when something activates it, the virus comes out, travels that path, and gets exposed again. There tends to be 4 things that bring out the canker sores in the mouth. If you get those, 98% of people have some type of herpes. Okay? It's a common thing. Most people get it to children and it just lasts for your life. 4 things that bring it out are peanuts, chocolate, stress, and sunlight. Don't think of the 4th one. Okay? We never have sunlight in the desert, and none of our students are ever stressed. Right? So chocolate, stress, peanuts, and sunlight are the 4 things that tend to bring out the herpes, the virus, and get your cold sores. So we do some, and there's ways to treat those. I know they're real painful. When you get a primary case, it's extremely painful, especially as a little kid. They won't eat, they won't drink. My dental assistant's kid had it when she was about 3 years old, and she would not eat or drink anything. Now, on my office, I have a laser. Put a laser on low intensity, low wattage, hold it just over the canker sore or the primary site of the herpes, and go back and forth about a minute. Don't ever touch it. It's completely painless. And it will seal over that herpes and it'll help make it disappear. And after, actually, they've been finding out, like with AIDS patients, they have a lot of active ulcers, a lot of stress to their body system. We get a lot of these, after about 8 to 12 treatments, the virus doesn't tend to come back in for a while. It's still been short-term studies, but it tends to be very good. So if we have a laser, we take the laser, we laser over it, and the pain goes away. And you're able to eat, able to drink, and it's great for little kids that get it. Because little kids, you can't put medication on there to seal it because they don't let you do that. All right, so those are the astrocytes. Astrocytes maintain the blood-brain barrier. Blood-brain barrier, this is an experiment done for penetration of morphine, codeine, and heroin. Okay? They took a rat, they gave it a bolus of codeine, heroin, and methadone into the carotid artery. They waited 15 seconds and cut off its head. Okay, very humane way to do things. Okay, so that stopped the distribution of it. So the uptake after 15 seconds of morphine was below measurability. The uptake of codeine was 24%. So codeine was 24%. Where am I at? Okay. 24% heroin, 16%, and methadone, 42%. So heroin, 68%, and methadone, 42%. Okay. Now, the funny thing is, okay, heroin cannot penetrate the blood-brain barrier. So it can't get into the brain. Heroin is converted into morphine, and then morphine penetrates the barrier. Okay. That's what allows that to take. So heroin has no effect on the brain. Heroin is converted to morphine to pass the blood-brain barrier, and that allows it to get in. Okay. That's why heroin is so addicting. Okay. because it can pass through that and then turns into morphine. So in other words, what happens is morphine is injected into your body and has a hard time going through the blood-brain barrier. It gets in about 24% of it. Okay? Heroin can pass the blood-brain barrier very quickly, its structure. In the brain, it converts to morphine, and then it has its effects. Because we have receptors in our brain that respond to morphine. We do not have receptors that respond to heroin. Okay? Heroin passes the blood-brain barrier more effectively than morphine. Once it's in the brain, it converts to morphine, and then morphine has the effect on the body. That's what makes heroin much more dangerous or much more addicting, because it has a faster effect, almost 3 times. Okay, 24% to 68%. It's almost 3 times the effect, okay, of codeine or morphine. Okay, so heroin passes the blood-brain barrier extremely fast. Then once it's in the brain, it converts to morphine. Morphine sits in the body. It can't penetrate the blood-brain barrier very quickly, so it has less of an effect. So heroin is much more addicting just because of the blood-brain barrier. Okay. Other neuroglial central system, oligodendrocytes we talked about. Oligodendrocytes have a lot of cytoplasmic extensions. Content the somas or the other axons. These extensions tie axons together in a sheath of myelin. They produce this covering or this coating over the axon that allows the nerve impulse to travel extremely fast. That's called myelination. Okay. So it produces these sheaths of myelin. Microglia cells are your phagocytes. Okay, they protect the neuron, they dissolve waste, they dissolve debris, pathogens, bacteria, viruses, whatever's there that's on there, these guys eat it, destroy it. That's your microglia. Okay, the fourth one is the ependymal cells. These are the ones that line the ventricles of the brain and they monitor your cerebral spinal fluid. So ependymal cells monitor cerebral spinal fluid and make sure that composition of that, the ions, the sodium, potassium, and the nutrients all maintain the balance that we need for correct functioning of the neurons. Right? And some of these actually secrete cerebral spinal fluid when it gets low. If our cerebral spinal fluid gets low, some of these cells, the ependymal cells, will create more and secrete cerebral spinal fluid back into the area. This is going to be your subarachnoid space we'll talk about in a little bit. Okay. And this shows you what it looks like. These are your ependymal cells. These are your astrocytes. These are your microglial cells. And these are your oligodendrocytes. That's where they live. That's where they function. Okay. We're going to talk about different matters in the brain of gray matter and white matter. Basically, gray matter is your cell bodies. That's what makes up the gray matter of the brain. The white matter of the brain is your nerve tracts. That's myelin. Those are your myelinations that you're going to talk about, which codes the axons. So we talk about gray matter. It's gray because it's made up of these cell bodies. It has mitochondria, it has endoplasmic reticulum stuff that gives it a darker color. That gives you the gray portion of the brain. The white portion is all these fat myelination coatings around the axon fibers that make it appear white. Okay, so here's your gray matter. When you look at a cross section through your spinal cord, here's your gray matter, which is your cell bodies, your white matter, which is your nerve tracts, your axons that transmit information up and down. Okay? And the central canal is the central portion of your nervous system where the cerebral spinal fluid runs to help maintain the environment. Okay, and you see ependymal cells here in this histological slide. They're the ones that monitor the cerebral spinal fluid. And this central canal here is filled with cerebral spinal fluid. And the ependymal cells that surround it can produce more cerebral spinal if it's low, can maintain the different ion contents and things that are required for the nerve cells to function correctly. All right, peripheral nervous system. Any questions on the central nervous system of the neuroglial cells? Everybody understands it. Good. There are 2 cells in the peripheral nervous system. Satellite cells and Schwann cells. Satellite cells regulate the exchange material between the cell body and the environment. Okay? They provide nourishment to the cells, get rid of waste, make sure that extracellular fluid maintains the correct balance so diffusion can occur, both oxygen in and carbon dioxide out, waste products out. Schwann cells, okay, they form the myelin sheath. These are the ones that create the myelin we talked about for the peripheral nervous system. A myelin sheath, if you have an axon, so here's your cell, and here's your axon coming through it. Okay, this nerve impulse travels. It's an electrical impulse that travels along that axon. If it's just a bare axon, there's a certain speed it travels at. If you take this and insulate it and wrap this around in this layer of a lipid, it's called myelin. Okay, if it's myelinated, it travels extremely fast because it jumps from area to area. It doesn't have to go through each section. It travels over the sheath and goes node to node to node. So it's almost, I think it's like 140 times faster. We'll give you the time to just a little bit here. If you have myelin around it versus no myelin around it. It's extremely quick with myelin. And that's what Schwann cells do. And that allows them to have the ability to help with repair damaged nerve tissue. Because it can produce these areas where these nerve axons can grow into and help repair damaged nerves. Okay. Here's your nerve cell body. This is your soma. Remember that? Has a nucleus inside it. You can see the satellite cells, these little cells that surround it right here. Okay. And those help nourish it, maintain everything, and connective tissue supporting the area. This is what myelin looks like. These are your myelin sheath, concentric rings that go around it. And you see how there's spaces in between the myelin. Those are called nodes. Nodes of Ranvier, actually, but they're called nodes. And allows the impulse or the electrical signal to jump from node to node to node. So it doesn't have to be this continuous signal that passes along the axon. Now it can jump extremely fast. You can go from node to node rather than travel a whole distance. It allows it to travel so much faster. So myelinated axons, myelinated nerve cells have an extremely fast impulse. Neurons themselves, we talked about the neuroglial cells, which are the supporting cells of the nervous system. Now we're going to talk about the nerves themselves, the neurons themselves, which transmit the information. So neurons consist of axons, soma, dendrites, terminal arborizations, terminal buttons, axon helix, where everything comes into it, and axoplasm, which is your cytoplasm for a neuron. So here's your nerve cell, here's your neuron. This is a helix right here. This is the area where the axon actually starts to begin and then narrows down. It's called a helix. Okay, these are your terminal buttons where the neuron ends and forms a synapse with another cell, another nerve cell. And this is the post-synaptic cell. This is the information where it's being received at, the cell that's being affected. The soma is your cell body. Remember, it contains of a nucleus, a nucleolus, which is just the center of the nucleus, ribosomes. Okay? That's what creates your gray matter, which gives you that gray appearance, that darker color, because they're chromophilic, ribosomes and neurons. Mitochondria to provide energy. Golgi apparatus to package the proteins being produced. They lack centrosomes. Okay, there are no centrosomes in a neuron. And what does that mean? That means the DNA cannot line up and be positioned and then be pulled apart to duplicate. So nerve cells cannot reproduce. The neuroglial cells can, but the neurons cannot reproduce. You are born with as many nerve cells as you're going to have. So every time you go on an alcohol binge or whatever, you destroy nerve cells, you don't get those back. And it's cumulative. Okay? That effect adds up over time. So again, here's your helix right here, beginning of where the axon is. You have your axon. Okay, the cell membrane surrounds it, cell body, your soma. You have microtubules, microfilaments that support the cells, just like we always do. We have the mitochondria. The main function of mitochondria is to produce ATP, produce energy. Cytoplasm or exoplasm. You have dendrites that feed information into the soma before voting on it, making a threshold, and sending information out through the axon. You got the nucleus, you got the nuclear pores, again, the same thing we've always had. So DNA can make RNA come out, make proteins. Okay. Now the nuclear membrane, the nucleolus, the Golgi apparatus, again, for modifying and packaging proteins. Rough endoplasmic reticulum, that's what the ribosomes are attached to. And you have free-flowing ribosomes also. So a lot of things we talked about before with a typical type of cell, except it lacks centrioles. It cannot reproduce. All right, so the structure of the neuron, we're talking about the axon. The axon has several parts. You have the axon hillock area, where the axon develops or is created from the cell body and creates an axon. The axoplasma is the cytoplasm inside, and the axon vesicles. Those are your neurotransmitters. Neurotransmitters allow the signal to transmit from one cell to another. So if this is your axon coming down here, there's your axon, and here's your cell. Okay, there's a space right here. This is called a cleft, or your neural synapse. So this is your synapse or your cleft, and material neurotransmitters leave the end of the neuron once it's stimulated and bind to receptors on the postsynaptic cell. And those trigger this cell to create another action potential. Okay, so that's how it's passed along. This is your synaptic junction. This is where the buttons are, where it ends. Okay, there's different types of synapses we're going to go over too. Okay. So neurons can be classified in different ways by structure or function. Structural classification. The structure is based on the number of processes extending from the cell body. Okay, how many dendrites you have. And some cells can have multiple axons too that extend from it. There's all kinds of variations. We're just talking about the basic structure first here. So the functional classification can be sensory, could be motor, could be interneuron. Interneurons are both sensory and motor, can carry both information. So sensory, is sensory afferent or efferent? Anyone remember? Everybody's sleepy today. Sensory is afferent. Motor is efferent. Remember, afferent is moving towards the central nervous system. Efferent is moving away from the central nervous system. Okay. Other structure way can be anaxonic, which has many processes but cannot differ between axons and dendrites. So it has all these processes, and the dendrites and axons look the same. So you can't tell the difference. Okay, that's anaxonic. Okay? And you only get those within the central nervous system. Could be bipolar. Doesn't mean it has a personality disorder. Okay, they're bipolar neurons. Okay? The cell bodies between the dendrite and the axon. In bipolar axons and bipolar neurons, the axons are not myelinated. They're slower responding neurons because they don't have myelination. Okay. There's pseudo-unipolar, where the cell body is off to one side of the axon. And there's multipolar, where single axon and multiple dendrites occur. This is the most common type we have in the central nervous system. That's our typical cell. All these dendrites receive information, go into the cell body, the soma, vote on it, reaches a threshold level, and then axon fires. Okay? That's the most common type that we have, multipolar. You have classifications on their function. Again, sensory, which is afferent. Okay, motor, which is efferent. Afferent is towards the central nervous system. Efferent is away from the central nervous system. Okay? For the afferent division, they're almost all pseudo-unipolar neurons. They send information from the peripheral nervous system to the central nervous system. Remember, there's somatic sensory And there's visceral sensory. Somatic sensory is information from your muscles, information from your glands. Visceral is information from your organs, your smooth muscle, cardiac muscle. Motor is efferent, going away from the central nervous system. So it sends information from your central nervous system to the periphery. And again, you have two divisions. You have somatic, which you talked about, and autonomic. Somatic. Okay. Somatic is what? Somatic is information going to the skeletal muscles to cause contraction, to cause an action. Autonomic is going to the parasympathetic nervous system or the sympathetic nervous system, where you're going to get ready for stress or you're ready to relax. The parasympathetic nervous system is a relaxful, peaceful part of your nervous system. The sympathetic is your fight response. Your body gets all keyed up. Okay? You can have interneurons. Interneurons are only found in the central nervous system. Okay, they're situated between motor and sensory neurons. They analyze sensory input and coordinate motor output. Okay? So information can come in from the afferent system, hit an interneuron. The interneuron can decide what to do. It doesn't have to go to the brain now. So the interneuron decides, and it kicks it back out to the motor neuron, and you get a response. Okay? It can be excitatory or inhibitory. It can cause an action to occur or can stop an action from occurring. If you put your hand on a hot stove, what happens? Move your hand, right? Now, the time it takes for that hand to move, if you measure the time it takes for the impulse to travel from your hand, up your arm, into the central nervous system, to your brain to go, it's hot, information goes back down, goes your hand moves, that's too slow. Your hand moves before that can occur. There's relays we found in reflex actions. That information goes from your hand, okay, to the spinal cord. The spinal cord has interneurons. Those interneurons say, move your hand now. So the information doesn't go to your brain. It goes down back to your arm and you move your hand. Your hand moves faster off a hot object or something sharp that's going to cut it or do damage than it is for that impulse to travel. And those are reflexes. It moves extremely fast. That's what you ever grab something really hot and you move your hand real quick. It doesn't hurt for a while, right? And after a while, after you move your hand and you realize, hey, that was really hot, and your brain starts thinking about it, you go, wow, that really hurts. Okay? So it takes off your brain to catch up because response is done through these interneurons in the vertebral column, in the spinal column. Okay? It happens in your spinal cord. It doesn't go to the brain for that reaction to occur. It goes on later on. After a while, it goes up to the brain. But the initial response doesn't get there. You move your hand way too fast for the information to go to the brain, think about it, and then move your hand. That's part of the function of these interneurons. Okay, there's three main types of receptors that are associated with sensory neurons. So sensory is afferent, information coming towards or into the central nervous system. Okay, you have exteroceptors, you have proprioceptors, and you have interoceptors. Okay, exteroceptors provide information about the external environment, like touch, temperature, pressure, sight, smell, hearing. Okay, so exteroceptors are active without you thinking about it. If you're sitting here watching me lecture to you, and one of your kids walk in behind you and drops a glass and it shatters behind you, what's your head going to do? It's going to turn behind you easily. You're not going to think about it. That's exteroceptors coming in. That's your external environment saying something's wrong, something's not right, and your head turns toward that automatically. You're at a restaurant, eating dinner, a waitress drops a bunch of dishes on the floor, you instantly turn that direction. That's exteroceptors that recognize your external environment. Okay, you recognize touch, temperature, things like that. Okay, that's exteroceptors. Proprioceptors. Proprioceptors is the realization of knowing where your body is at all times. If you stick your arm out, you know where your arm is, right? And not because you can see it. Close your eyes and stick your arm out. You know where your arm is. That's proprioception. That's the ability of the body to monitor the position and movement of your body, to know where all things are within your body. When we do denture cases for people, take the teeth out and put in a denture. If the teeth are really bad, okay, they lose proprioception in the mouth because all the nerves are covered now. Because you have proprioception where food is when you're chewing. You don't think about, oh, let me move this piece of food unless you got a toothache, then you realize you can't chew on one certain side. But you have proprioception. Your mouth and the neurons know where the food is located in your mouth, so you can chew it and move it around without thinking about it. When you cover over those nerves, you don't have that anymore. So you lose proprioception when a denture covers over it. Okay, so you have exteroceptors, you have proprioceptors, and you have interoceptors. Interoceptors monitor your internal organ activities. Digestion, you don't have to think about peristaltic motion through your digestive tract. Your body does it automatically through interoceptors. It monitors your internal organ activity and makes adjustments to correct everything. Okay? So those are three major types of receptors that you have for sensory input, for efferent operations, where everything's coming into or towards the central nervous system. So exteroceptors, proprioceptors, and interoceptors. Different functions. Okay. And here's another diagram of the different types that you have. Okay. You have your receptors, which you talked about. Those are three different types. Okay. They feed into the afferent system in your central nervous system. Your central nervous system decides what to do with it. What is a somatic response, which is muscular. The skeletal muscles contract. Okay. These are inner nerves. Or it can come into visceral. Visceral is your smooth muscle, your glands, cardiac muscles. And it can come into here and feed it that way. Okay. And it says here, we haven't gotten to this yet. These are preganglionic fibers and postganglionic fibers. Each in a ganglion. A ganglion is by definition a group of nerve cells that are together in a swelled area or just a real compact area. It uses cause like a little swelled branching inside the nerve tract. That's a ganglion. And you have fibers before and fibers after. That's where the nerve cells are located, where everything votes on and gets sent on down. Okay. Neuroregeneration. This is what we talked about. We have limited ability to repair nerve tissue when it's damaged. If you completely sever a nerve, there is very little chance that that nerve will repair itself. If you crush or damage or kind of tear a nerve, there's a chance you can have the limited ability to repair it. And again, you can never grow more neurons. You're born with as many as you can have. So what happens when you have limited repair, you damage or crush the nerve. Okay? Schwann cells grow into the cut area. Remember Schwann cells? Schwann cells produce myelination, it helps surround the cells, and allow that impulse to continue quickly. So the axon sends buzz into the network of these Schwann cells. So these Schwann cells create this network. Okay, you create all these networks right here and give this great area. So the neuron can extend into that area now with the myelin surrounding it and helps push it into the new area. So the axon grows into these Schwann cells. Okay, and it can reestablish connection with the damaged area. So these Schwann cells aid in helping to repair. I get limited ability. This isn't regeneration. This isn't great repair. This is a limited ability to repair Schwann cells. Just like muscles have some limited ability with the myosatellite cells we talked about. Okay. Now bone, on the other hand, bone has extensive ability to repair. You can always repair bone. That repair is tremendous. You can break that into pieces, into multiple pieces, and still repair it. Okay. The nerve cells, very limited repair. And here's how it works. Okay. This is your distal stump. This is the area that's been damaged right here. Okay. You have Schwann cells that grow into this area. These are your Schwann cells here. And they start to form this matrix. And they start to form this area that this nerve can now extend into. And then as the nerve extends into that area, it can reestablish a connection inside where those areas are being protected. And that's how you get limited ability to repair itself. You do not regenerate. You do not make another neuron right here. Okay, that does not happen. But you can extend your existing neuron into the damaged area with the support of these Schwann cells and reestablish a connection. Is that making sense? Okay. I don't want to give you the idea that neurons can regenerate. They can't. But you do have limited ability to repair because of these Schwann cells creating an environment where you have a matrix that the neuron can grow into, the existing neuron, and reestablish a partial connection. Okay? The nerve impulse itself, the impulse is an electrical current that travels through your body. It's made by the exchange of ions, potassium and sodium. They change places. Potassium ions go out, sodium ions come into the cell. Okay, that creates an electrical potential difference because charges are being moved and the electricity is just a charge. And it takes that current and sends it down the nerve. So a nerve impulse is what we call an action potential. Okay, so action potential, here's your axon. Okay, and the action potential is a spike. Okay, difference of potassium going out, sodium coming in, change of current. That's your action potential. Action potential is used to the exchange of ions, sodium and potassium, across the membrane. And we didn't talk about the sodium potassium pump yet. That was physiology. We will talk about the sodium potassium pump to pump the ions across again so the nerve can fire multiple times. So it's the ability to conduct the impulse is known as excitability. The nerve can be excited and create an action potential, which shoots an electrical signal down the nerve. Okay? So if we block the ability of sodium and potassium to move, so if we stop potassium and sodium from changing places, okay, if we prohibit that, the nerve will not fire. That's a nerve block, what you do on a patient. You do surgery when you have I had a ganglion cyst removed from the back of my hand. They block the nerve. They can block it up at the elbow. They can block it at the wrist, but they can block the nerve. If you stop that nerve from firing, you don't feel pain. The pain's still there. The nerve is not firing because you're blocking the sodium potassium pump. You're not allowing those ions to move. So all I'm doing when I do dentistry, I'm not taking away the pain. When I give an injection, all I do is stop salt from moving. That's what local anesthetic is. It stops salt. The sodium, potassium, or salt, they just change place. So all I'm doing is stopping salt, and that nerve can't fire. If the nerve can't fire, you can't tell your brain you're feeling pain. That's how it works. So that's an action potential, is the exchange of ions, and it's called excitability. Excitability is the ability of the nerve to fire an electrical impulse, which is an action potential. Okay. The stimulus. A stimulus is something that causes an action potential to happen. Stimulus could be a needle prick. Could be a hammer hitting your hand with a hammer when you try to nail the wall, right? That's a stimulus. It's something that triggers a nerve to fire. Yeah, it doesn't have to be extreme. I mean, you can put your hand on the desk and you can feel it. You can pick up a pencil and you can feel it. Those are stimulus. You get stimulus all the time from input from your afferent nervous system. And it causes an action potential to occur so that information is traveled to your brain. Okay? If you put a bowl in the microwave and you grab that bowl and it's hot, okay, that's a stimulus. It tells your brain, don't do that. Don't squeeze around it. Okay. Now, some of the things with stimulus can be really tricky, like an electrical fence. If you ever worked on a farm, worked on an area around horses or cattle, where you put up electrical fences, the way you test an electrical fence is by doing what? You test it with the back of your hand because it's an electrical signal. Remember what your nerves are. Your nerves are electrical signals. So if you grab an electrical fence with your open palm, it's going to contract your nerves and the muscle is going to contract. What's your hand going to do? It's going to clench. It's going to clench your electrical fence and you're going to electrocute it. Then he's going to send those electrons through your body. You're not going to be able to open your hand because it's an electrical signal. An action potential is an electrical signal, and it's a stimulus. Okay? So if you touch it with the back of your hand, if that fence is live or hot, your muscles are going to contract, and it's going to pull your hand away from the wire. So there you have a life lesson there. If you want to test electrical fences, always test with the back of your hand because electrical signals cause your muscles to contract. The same thing in your body. They're electrical signals. Okay? So that's a stimulus. Anything that causes an action potential, which is a spike, electrical signal to occur, is a stimulus. The stimulus has to reach a threshold maximum for that particular neuron. Okay? If you grab something that's room temperature, you're not going to get a heat signature through your threshold. It didn't reach a threshold. If you grab an ice cube and squeeze it, that's going to create a threshold. It's going to be too cold, and your body's going to know it. Once it reaches that threshold, it causes that nerve to fire. You grab something too hot, it's going to reach that threshold, cause that nerve to fire, cause that action potential to occur. So the threshold level is the amount of stimuli required to create an action potential. Okay? That's how much you need to make that nerve fire. Okay? It has to reach a certain level to let your body know it's too hot. It's too cold. Okay? It's too sharp. It's hitting me too hard. It has to let you know, it has to reach that level, that threshold level. And once you reach that threshold level, it's going to fire. I mean, you can take a tooth and tap on it, and nothing's going to happen. You didn't fire the nerve. I can take a drill and tap on it with the drill running, and you're going to feel it. Okay? It passes that threshold level for the stimulus. Once an impulse starts, it propagates along the length of the axon. Important point. Okay? Once the impulse is triggered, there ain't no way to stop it. It's going the whole way. It's either on or off. There is no, we're going to contract a little bit here. Okay? It either goes or doesn't go. Now, multiple neurons can be involved. So if you're not contracting fully, not all the neurons are firing, which is fine. But each individual neuron for the axon, once that stimulus causes a threshold level for that axon to cause an action potential, that action potential fires 100%, goes all the way across. You can't stop it once it starts. Like it's an on-off switch. It goes, it doesn't, it doesn't. There is no midway on these things. I used to work as cutting lumber, cutting trees down up in the Plumas National Forest. And if you ever work with these guys, like they're really good lumberjacks, they cut some big trees down, and you realize once you reach a certain point and that tree starts to move, there is no force on heaven or earth that you can stop that tree from moving. No matter what's in the way, it's amazing the power those things have when those trees come down. You know, it's like an action potential. Once it starts, it's going, and you're not going to stop it. Okay. So once this impulse starts, it goes. Okay. Once it reaches that threshold level, it triggers it, and there is no maybe about it. You have a threshold level, it hits it, and it travels. 100% goes all the way. All right. The speed we talked about. We talked about a really important myelin sheath produced by the Schwann cells, remember? So the speed of the impulse depends upon two factors, basically. The diameter of the neuron and the presence or absence of a myelin sheath. Okay? If you have the presence of a myelin sheath, it's an extremely fast impulse. Lack of a myelin sheath is a slow impulse. Okay. Myelinated axons with a large diameter, okay, 140 meters a second. It's fast. Okay. 140 meters every second. Unmyelinated and a small diameter, less than 1 meter per second. So with myelin, the large nerve, it's traveling over 140 times faster when myelin is present because it's jumping from node to node. It doesn't have to travel the whole length of each step. It catches this node, this node, this node. So it jumps. If you're not making that physical connection all the time, it can go a whole lot faster, at least 140 times faster. So two things affect it: larger diameter, which is very much affected because it's bigger surface area, more to travel with, and the presence of myelin. So far, so good. All right. Now we're going to talk about the synapse. Remember the synapse, okay, was the junction of one nerve to another nerve, or one nerve, the end of the nerve to a muscle or to a gland. It's where the nerve, the axon of the neuron ends and connects to something to cause an effect, whether it passes on to another neuron, whether it causes a muscle to contract, whether it causes a gland to secrete, whether it causes your heart rate to increase, whether it causes your pupils to dilate, pupils to contract, whatever it does, that's the effect it has. So a synapse is a junction between the neuron, the axon of the neuron, and its effector, what it's going to affect. Okay? There's different types of synapses. Okay? You get an axodendritic synapse, which is the axon of one neuron, affects or hits the dendrite of another and feeds information into it. That's one of our typical ones. And then when enough information comes in the dendrites, the other axon fires. You can have an axosomic synapse, where the axon of the neuron hits the soma or the cell body of the next neuron, okay, and affects it that way. Or you have an axonic synapse, where the axon of the neuron hits the axon of another neuron. So the axon can hit the dendrite, the axon can hit the cell body, or the axon can hit another axon. Those are all different types of synapses that can occur. Again, axodendritic, axosomic, or axonic. Okay, the synapse is the junction, okay, between the neuromuscular or the neuroglandular. Okay? So the axon of a neuron on a muscle is your neuromuscular junction. When the neuron, or the axon of the neuron, terminates or hits a muscle. It is only when a neuron hits a muscle, there's only one thing it can do. It has to cause that muscle to contract. It can't do anything else. Okay, so once the axon fires and the ending point is the muscle, that muscle is going to start to contract. That's its only effect. Neuroglandular is when the axon of the nerve ends on a gland. So the axon of the nerve goes to a gland. And the gland does a secretion, whatever it's going to feed, whether it's the pituitary gland, whether it's sweat glands, whether it's holocrine glands, American glands, whatever it's going to affect, it affects that gland and causes its secretion. So you can have neuromuscular or neuroglandular synapses also. Okay. And here's some of the different types. This is synapse with another neuron. In other words, this acts, here's your axon, okay? And it feeds into, right here it's feeding into a dendrite, right here it's feeding into the cell body. Those are two different types. Now, if it fed into the other axon, it would be the third type, axoaxonic. Okay? So here's your fire. This is your action potential. This is your stimulus, comes in, causes nerve to fire. You get your action potential. It travels down the axon. And right here it goes to dendrite. And right here it goes to the cell body. And both it feeds information. When the threshold level is reached from that information coming in, then this axon fires, but not until. And once it hits that threshold level, it's 100. It fire. Like I said, there's no little peaks. It either fires or it doesn't. It fires at 100% capacity once it reaches threshold. Once you pass threshold, that thing goes. There is no moderation. There is no on-off. So it's an on or off neuron. It either fires or it doesn't. There's no in between. Neuromuscular synapse. This is where your nerve ends. Your neuron ends on a muscle. Okay? So the ending of it goes to the muscle and it causes that muscle to contract. That's your neuromuscular synapse. Neuroglandular synapse is when the neuron ends on a gland. Okay? And these has a synapse, causes the neurotransmitters to go across that space and causes that gland to do its function, to secrete whatever's inside of it, depending on what the gland is. Okay? So neurons can innervate different things depending on where we're looking. Okay, we still got good timing. Okay, synaptic communication. There's two basic types or ways a synapse can be used. Okay, so the synaptic terminal, the nerve impulse triggers events at the synapse that transfers information across that synapse. The synapse is a space. So this is your axon coming down here. This is your axon. You had your threshold. The response comes down here. Okay? And inside of here, these are being released. And there's little receptors here. And the receptors bind to it. Okay? So a synapse is where information is transferred from one neuron to another neuron to muscle. Is information being transferred across that space? Okay? There's two ways to accomplish it. A vesicular synapse, which involves a neurotransmitter, which means acetylcholine is released, another neurotransmitter, and it causes the other nerve to fire or have an effect if it reaches the threshold level. Okay? So there's vesicular and there's non-vesicular. Non-vesicular means ions flow across, sodium, potassium, These ions flow across, and those ions act as transferring information. Now, the big difference, which will appear on your tests, is vesicular synapses can only go in one direction. One cell has a neurotransmitter, one cell has the receptors. That information travels unilaterally. It can only go one way. A non-vesicular synapse, ions can infuse both directions because all it is transferring ions from one membrane to another. So the ions can go left to right, and then go right to left. Okay? So vesicular synapses are one-way streets. They can only transfer information one direction. Non-vesicular synapses are two-way streets. Information can go from the first one to the second one, or from the second one back to the first one. So it's able to take information both ways. Because non-vesicular is a flow of ions. Vesicular is the release of a neurotransmitter. A neurotransmitter has specific receptors that it binds to allow that information to be transferred. Okay? Whereas ions just fuse across a membrane. They don't need receptors. Making sense so far? I'm not hearing any arguments so far.
Yes.
Okay, good. Sometimes I wonder if anybody's listening to me. It's hard to tell sometimes.
We're
Okay.
here.
Okay. So vesicular synapses, this is what occurs for vesicular. Remember, vesicular is one way. It's a neurotransmitter. It's being released. Okay. So the sequence of an axon potential arrives at the presynaptic membrane. The presynaptic membrane is a membrane before the synapse occurs. Okay, once that axon potential gets there, it is released by a neurotransmitter. The neurotransmitter for the axon travels into the cleft, into the synapse. So it diffuses across the synapse. It binds to receptors on the postsynaptic membrane, the membrane on the other side. Has receptors that the neurotransmitter fits into that are very stereospecific. It binds to that, and now that activates the postsynaptic membrane. So your binding axon causes a change in permeability of the postsynaptic membrane. That change in permeability results in an axon potential of the next neuron, or contraction of the muscle or the gland, or whatever it's ending up at, wherever it's hitting. That's your synapse events. That's what happens. The neurotransmitter, the axon potential travels down the presynaptic membrane, because if you have two muscle neurons here, okay? This is the presynaptic before the synapse. This is the post. Remember, this is going to go one way. This is a vesicular event. So the presynaptic membrane comes down, releases neurotransmitters. They go across. They bind to the little receptors here. Okay, cause a change in permeability. Another axon potential. So you've got a presynaptic membrane and a postsynaptic membrane. This is for vesicular because it can't go both ways. You have to have a pre and a post. It only travels from the presynaptic membrane to the postsynaptic membrane. That's all it can do. So here's what we look like. This is our soma or cell body. These are all the dendrites of a typical neuron. And this is the axon. What are all these little sausages here? What's that called? Called myelin, right. That wraps around and has it faster. What are these little areas called between the myelin, like these spaces?
Nodes of Ranvier.
Nodes of Ranvier. Those are your nodes. That allows your impulse to skip along extremely fast down this thing. Okay? Then it comes to an end where you have your terminal buttons. You see your terminal buttons here, where they're binding onto other dendrites. This is your presynaptic membrane. These ones here are your postsynaptic membrane. Okay? And what happens is the nerve impulse comes down, in this big diagram right here. You can see all these little neurotransmitters, and they get released into this cleft right here between the cell membrane and the neuron. They travel across the cleft to the postsynaptic membrane, bind to the receptors, cause a change in permeability, cause another axon potential to occur. And then now the information is traveled along and passed along. Non-vesicular events. Non-vesicular events, remember, can go both directions because all it is, there's no neurotransmitter, there's no receptors. It's just ions moving. And ions can transmit in passive diffusion, can go by active diffusion across membranes. And they don't need specific receptors, so it can travel both directions. So you have a presynaptic membrane, and one neuron is tightly bound to the postsynaptic membrane of the other one, and ions can pass from one neuron to the next, and they can exchange either direction. So remember, vesicular events, one way. Non-vesicular events or ions, two ways. The organization of neurons, they organize in certain patterns of how they propagate information through the body. So they're organized into smaller organized groups called neuronal pools. Okay, it's not a swimming pool, but it's just a group organization of how they relate information under a certain pattern. Neuronal pools are identified by their neural circuitry. Just like electronics have different circuit patterns, so do our neuronal pools, because it's electrical. Our body runs on electricity. Okay, it's exchange of ions. It's just like current. We have different ones. We have divergence, we have convergence, serial processing, parallel processing, and reverberation. And I guarantee you're going to see this on the test. Okay, so it's something you should know. Okay. Divergence. Divergence is just like it sounds. The spread of information from one neuron to several. It diverges. Okay. Permits broad distribution of a specific input. So it allows one input to spread to many other areas. Okay. So information enters the central nervous system and then spreads through the brain and the spinal cord at the same time. So one stimuli can affect different areas of the spinal cord, go to the brain and the spinal cord simultaneously by divergence. It diverts the path it takes. So you can spread it to different areas, which is very advantageous. Convergence. Convergence is the opposite. Information going from several neurons to a single neuron. It converges. Okay? Movements of your diaphragm muscle are involuntary. You breathe without thinking about it. But yet at times we can move the diaphragm muscle voluntarily. We can force a breath. So we can take all those neurons that are going to the diaphragm and force it into a single area and make it fire. We can control it if we want to. We can't do a lot of other things. We can't control peristalsis. We can't control our heart rate by thinking about it. Right? So that's convergence. Okay? You will see pictures on your exam. If you see this picture, you will say divergent. One neuron is going into several different places. That's divergent. Divergence. Convergence. Several neurons are feeding into one. That's convergence. So far, so good? We're not too tricky here. Divergence and convergence. Serial processing. Serial processing is in series, like an electronic circuit. Its information goes from one neuron to the next in sequence. Bang, bang, bang. Just follows a sequence. Okay? So it goes from one to the other, to the other, to the other, all linear. So information going from one part of the brain, then to another part of the brain, then to another part of the brain. It just goes in a series. Okay? Just like electrical current. Parallel processing. Several neurons are processing at the same time. You have several serial processing. You have these one going in sequence. So you have one neuron goes to another, going to neuron, and one neuron going to another, going to another. So you have several of these going at the same time. When you step on a nail, it says you yell ouch. Trust me, people don't say ouch. If you step on a nail, other words come out. Okay? Why? How's that happen? You feel the pain and you say the word at the same time. How can that happen? That can happen because you have two parallel paths in serial processing. That's parallel processing. It's going to different places, but it's the same serial path. That same stimulus is affecting a different path simultaneously. So you can yell and cuss at the same time that you feel the pain. Okay? This is serial processing. This is a picture you're going to see on your exam. It's one neuron going to another neuron, going to another neuron in series. Just bang, 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 follows the path. One part of the brain, another part of the brain, another part of the brain. That impulse goes on. Okay? This is parallel processing. You get your stimulus here, you step on the nail, then you get this series again. This goes here, and it's parallel. It's different series going together at the same time. Okay? So if this goes to your mouth, you get to cuss. This goes to your brain, you get to feel pain. Okay? It goes to different areas, but it's a serial processing. That's called parallel processing because they're in parallel. They're all going the same direction at the same time. So now you got serial processing and parallel processing. Convergence, divergence, serial processing, and parallel processing. Okay, reverberation. Reverberation is the only one that's a little bit different. Reverberation is a feedback loop. Okay, it's collateral access and back to the original impulse that either enhances the pain or continues the pain. Okay, if you put your hand on a hot stove and you don't move it, it's going to keep hurting. It's not going to stop until you move it. Okay, it's going to keep telling you move your hand, and it goes through a feedback loop that says move it now. Okay, it's not going to just process it and go away. So this is reverberation. Here's your stimulus right here. This goes down, and it starts to go in here. If something doesn't stop to break this loop, it goes back around here, and it refires it again. It says, hey, pay attention to it. And it keeps accentuating or taking that mechanism and causing it to keep firing. Now, it can be excitatory, make it keep happening, or it can be an inhibition that you want the body to do. But it's a feedback loop. That's reverberation, reverberates into a loop. So those are the 5 different types you need to know. Okay. Let's finish this chapter, then we'll take a break. It'll only take another 5 minutes or so. Okay, so organization of the central nervous system. We talked about gray matter, we talked about white matter. Gray matter is your cell bodies. That's what they are. They're the endoplasmic reticulum and the Golgi apparatus have the proteins in it. Those are given its gray color. Okay, so the gray matter organization is your neural cortex. They are the surface on the brain. Okay, they're the cell bodies in the interior of the central nervous system. Your white matters are your bundles or your nerve tracts, where the information or the action to travel along. And the reason they're white is because myelin is white. It's like a fatty pad wrapped around it. So myelin's white, and those are myelinated nerves. So that's your white tracts. The bundles are either ascending or descending. Ascending is coming towards the central nervous system, which is afferent. Descending are going away from the central nervous system, which is efferent. Okay. So peripheral nervous system, gray matter, white matter. Okay, gray matter is your cell bodies, white matter is your nerve bundles. Okay, this is the neural cortex. This is where all your thinking goes on. Okay, it's gray matter on the surface. That's where the cell bodies are. And all this white matter is all the tracts that are coming at you and transferring information in. Okay, so the white matter is the tracts or bundles of the nervous system. They have they share a common destination. So a nerve tract is different areas that go to the same place in the brain. In the peripheral nervous system, same thing. Gray matters are ganglia. Remember, ganglia is a group of cell bodies. So ganglia is good gray matter. They're your neuron cell bodies. White matter is bundles of axons, myelination. Okay? Again, you have the gray matter, white matter. All right, who's paying attention? Number 1 is what?
Divergent.
Divergence. Perfect. You need to know this for exam, so pay attention to it. Number 2 is what?
Convergent.
Convergence. Number 3 is?
Serial.
Serial processing. Number 4 is?
Parallel.
Processing.
Perfect. Parallel processing. And number 5 is?
Feedback loop. I don't remember what it's called, but it's the last one.
Reverberation.
Reverberation. There you go. Perfect.